how we understand how they actually work, is through crystallography that's been done. So people have been able to crystallize the subunits of the ribosome, or in some cases the whole ribosome with both subunits there, with inhibitor molecules bound with various kinds of antibiotics. And so we know now how many of those drugs actually work. And this is a picture taken from the website of Venki Ramakrishnan, who is a Nobel laureate who solved the crystal structure of the small subunit of the bacterial ribosome. This is the 30S subunit. And what this is showing, and it's sort of, this is such a beautiful big molecule, it's actually hard to do it justice here, but so it's mostly RNA, right? It's about 2/3 RNA by mass, about 1/3 protein in this big complex. And this tiny molecule that is an antibiotic actually binds right in the heart of the real business part of the small subunit, and basically is able to block this subunit's ability to associate with substrates. So it functions as a competitive inhibitor of the ribosome, and makes it difficult or impossible for this subunit then to function in protein synthesis. And the reason that this works and it's not toxic to us to take drugs like this is that these molecules tend to be very selective for bacterial ribosomes, and they don't tend to bind well or inhibit mammalian or human ribosomes. So there's some interesting chemical differences in those subunits that allow these molecules to work on bacterial ribosomes and not on humans, thank goodness. Ribosomes are all made of RNA plus protein, but there are differences in the structures that allow these chemicals to interact very selectively. Remember that enzymes are super selective for the kinds of molecules and substrates that they bind to. Now, this is a cartoon that shows sort of diagrams competitive inhibition and, in this case, non-competitive inhibition. And as we said before, we can think about the enzyme as having, of course, it has an active site, it has a substrate binding site, but it might have other nooks and crannies, it might have other pockets that are accessible to small molecules. And so when we have a competitive inhibitor, we have something that looks enough like the substrate that it can actually interact and directly compete for binding with the substrate. Whereas if we have a non-competitive inhibitor, this molecule might look very different from the substrate for this enzyme, but nonetheless, it has the ability to associate potentially very selectively with some other site on the enzyme. And when that happens, it might cause a conformational change in the protein that prevents it from binding to the substrate, possibly, or might still allow it to bind to the substrate, but not in an orientation that is productive for chemistry. Okay, so that's the difference between these kinds of inhibitors. And then I told you that I was going to tell you about an experiment that was done with the ribozyme system that we were studying in my laboratory to figure out whether this metal ion cofactor was really essential for the chemistry of the reaction. And so we did an experiment in which we introduced a molecule called cobalt hexamine into the solution that was used to crystallize this RNA. And so what we could do is then solve a crystal structure with this molecule bound. And the reason that this was a good way to test the hypothesis that the magnesium ion observed in that original structure of the active form of the ribozyme, to test whether that was actually necessary for the chemical reaction catalyzed by this molecule, is that it turns out that biochemically, if you take a purified mixed solution of this ribozyme molecule, and you add cobalt hexamine to that reaction, there's very strong inhibition of the reaction. Okay, very strong inhibition, and that's shown in a plot over here. So if you do what's called a double reciprocal plot, where we plot 1 over substrate concentration on the x-axis and 1 over velocity of the reaction on the y-axis, this is a very typical pattern that you observe as the concentration of a competitive inhibitor increases, where the velocity of the reaction does not change, but the effective concentration of substrate that's necessary to get to that maximum velocity increases. Okay, and so this is what we observed experimentally when you add cobalt hexamine to this ribozyme reaction. And so then when we did the crystallography, what we saw was that there was electron density corresponding to the presence of this cobalt hexamine ion sitting right in the active site and almost exactly at the same location as the location of a magnesium divalent ion. And so that certainly was very strong suggestion or strong experimental evidence for the idea that this ion is actually functioning as a competitive inhibitor, competes for binding, in this case with this cofactor that's necessary for the reaction to occur, and prevents it from occurring efficiently. So let's try a couple of questions. So get out your clickers, and let's try a couple of questions on enzymes. Oh, how come I can't see my, oh no. Oh, there we go. Okay. It was going to be really a trick question. Okay, so let's just take a minute to do this, and I'm going to start the clock right now. Okay, so about 10 seconds to go. 4, 3, 2, 1, stop. Okay, so interesting distribution here. So 30% of you said that the answer was A. Okay, now that's what I would have said too. Okay, so I think that the answer is probably A, because we're saying that we have an enzyme catalyzed reaction that's going at this rate here. We add a substance to the reaction, we see a pretty strong reduction in the rate. And if we add more, it doesn't alter the rate. Okay, so I think that is probably most consistent with this being a competitive inhibitor of the enzyme. Now, a lot of you said D, and I'm looking for, where's Mike? What did you say, Mike? You said D. Okay, so, all right, so let's talk about this, because I think D is another possible answer here, and I'm going to give you credit if you said D, but I'm going to tell you why I don't think that's actually the most logical answer here. Logical, okay? And the reason is that this D says substance A induces a conformational change in the active site by binding elsewhere on the enzyme. Now, we talked in class about binding elsewhere on the enzyme being inhibitory, but it doesn't have to be, right? And there actually are interesting examples where you have molecules that will bind somewhere else on an enzyme, and they actually activate the enzyme. And we'll talk about that with Alice theory, okay? So I would argue that based on the data you have here, that's not necessarily the correct conclusion. Yes? Right. Well, you know, it's always tricky to, as you can see, it's tricky to word these questions so that everything's really unambiguous. Maybe what I should have said in the question was, you know, you have a giant excess of substance A, right? So that even if you add more of it, you know, nothing's going to happen because everything's already saturated. Okay, might have been a better way to word that question. And what was the first thing that you said? Oh, because it slows the reaction. Okay, but this is important, right? So this is an important point. So inhibition, by that word, you might think, oh, I mean that the enzyme actually stops and we don't see any further catalysis going on. That's actually often not the case. So what we typically see with enzyme inhibitors is that they slow the reaction, they don't stop the reaction. Again, remember that that's really because everything's in equilibrium, right? There's an equilibrium going on. So things are coming on and off. The inhibitor's coming on and off the enzyme, unless it's a suicide inhibitor, right? But that's a special case. But let's say for reversible inhibitors, they're coming on and off the enzyme, and the substrate is too. So there's this constant competition going on. And of course, everything will be affected by the relative affinity of the substrate versus the inhibitor, right? So yeah. Shout it out. What would be a question in which D was the correct answer? Well, I would just turn that around and say if in D I had said that this is that this binds somewhere else on the enzyme and inhibits the enzyme, then I think D would be an equally correct answer to A. Okay, if I'd worded it that way. Okay, yeah, question here.
So these enzymes are coming on and off, and the way they're slowly reacted by blocking other ones. And wouldn't increasing substrate always further slow down?
He's asking, wouldn't increasing substance A always slow down the reaction further? Only if it depends on two things. It depends on several things. It depends on the concentration of enzyme, the concentration of the substrate relative to the concentration of the inhibitor, and then the affinities of those two. Okay, so I'm giving credit for D because I understand why you might have chosen D here because there wasn't maybe enough information given to be absolutely sure that this was a competitive inhibitor. I would still say that I think the most logical answer is A. But yes. No. So she's saying so suppose I just gave you an enzyme catalyzed reaction, and then I gave you a tube of something, and I said just squirt it in and measure the rate, and then tell me what kind of inhibition we're observing. Okay. I would argue that there'd be no way. You could certainly tell me whether inhibition was going on, right? Because you're measuring the rate, you see it slowing down. But I would argue there's no way that you would know without knowing more information, like the structure of the thing that I have in solution I gave you, or something like that, whether it's a competitive inhibitor or whether it's some other kind of inhibitor, without more information. No, because there's not. It's not as though competitive inhibitors are always better, right? There's lots of great examples where they're non-competitive inhibitors that are still great inhibitors, right? Because they just bind really well, but they just happen to not compete with the substrate. Okay. So let's do one more question. So don't put your clicker away quite yet. Okay, so let's do this question here. Okay, let's take this is an easy one. Take about 40 seconds to do it. I'm starting the clock right now. Okay, about 15 seconds left. Okay, 5 more seconds. I'm stopping the clock. 2, 1, now. Okay, so, okay, that's great. So everybody said D. Okay, and I agree. I think it's D. So we agree. That's good. Right, and so again, this is just underscoring the point that enzymes are, they're either going to be proteins or RNA molecules, right? And as we talked about last time, they tend to be evolved to match the environment of the organism. So if an organism is growing in a high temperature environment, we find that the enzymes tend to be, evolved to function best near that temperature. Okay. All right, so let's move on. I want to just talk a little bit more, tell you a couple other things. So this is just kind of a really interesting example medically. So you probably heard about drugs that inhibit the HIV virus, the virus that causes AIDS. So this is an RNA virus. And it turns out that when HIV enters the cell, so can everybody see down here, hopefully? So when HIV goes into a cell, so it's an RNA virus, and it actually encodes, it has one long, what's called open reading frame. So it has one long sequence that encodes what's called a polyprotein. And so this RNA gets translated by the ribosome and that translation synthesizes a long polypeptide that's based on the coded sequence here in the virus. Now this polyprotein is not functional as a polyprotein. It actually has to be digested by one part of this protein that actually gets snipped off by an enzyme in the cell. So it is, there's some functional enzyme made called HIV protease. And this protease is present in very small quantities, but it's a catalyst, right? So it's enough to catalyze the breakage of bonds in this polypeptide to release the individual functional proteins that are necessary to make new HIV viruses. And so this is an essential part of the viral life cycle. And so one of the ways that researchers have found to inhibit this process, and it turns out that this is a very effective therapeutic for HIV, is to actually make a competitive inhibitor of this HIV protease. Oh dear, I just lost my pointer. But it's that molecule right there in the center. I'll use my pointer. It's this molecule right here, and this is a crystal structure of the protease enzyme. You can see that this molecule actually binds, and it prevents interaction of this enzyme with its polypeptide substrate. Yeah. Are enzymes ever terminated by the inhibitor? Well, I think you're talking about what we said about suicide inhibition, right? Where you have a covalent bond that's made, and the enzyme is effectively it's permanently inactivated. But that's not what's going on here. So all the inhibition that we're talking about, these examples in class, these are reversible inhibitors. Okay. So that's just one cool example. There's lots of other examples that are really important therapeutically. Thank you. Yay. All right, good. So we don't have too much time left, but I want to talk about allostery because this is a really important concept that to think about because it affects a lot of enzymes and other kinds of molecules in the way they function in the body. And so when we talk about allostery, we're really talking about interactions that occur somewhere on an enzyme, or very typically for allostery, we're talking about multi-protein complexes, like the example shown here. This is actually hemoglobin, which is not an enzyme, by the way. This example, it actually binds to oxygen. And allosteric regulation can either inhibit or it can stimulate enzymes or other molecules like this that are for binding. And this often occurs when a regulatory molecule binds to a protein at one site and it affects its function at some other site. Okay, I'm going to show you. We'll look very briefly at how this works for hemoglobin in just a moment. So these are typically made from multiple polypeptide subunits that come together. And for allosterically activated enzymes or inhibited enzymes, these have active and inactive forms. And what we find is that the binding of an activator will stabilize the structure of the active form, whereas binding of an inhibitor will stabilize the structure of the inactive form. Right? So you can sort of think of this as toggling between an active form and an inactive form. So here's some cartoons. These are actually from your book showing how we imagine that this works. And so if we have an allosteric enzyme that has 4 polypeptide subunits that come together to form an active or form a complex, if we have an activator, this molecule binds somewhere, not to the actual active site of this, but somewhere else that ends up stabilizing the active form of this complex. So it's an activator in that sense. And conversely, if we have an inhibitor, inhibitor, we have a molecule that binds, again, typically somewhere else on this complex, but stabilizes the inactive form of the complex. Okay? And so in this example, only molecules, only complexes that are stabilized in the active form like this are then able to bind substrates and catalyze reactions. Okay? So it's sort of analogous to the idea of non-competitive inhibition, where you have a molecule binding somewhere else, not in the actual substrate binding site, but affecting the ability of this enzyme complex to function. And this is just showing a close-up of that example. And I want to just talk quickly about hemoglobin, because this is a really classic example. This is kind of one of those examples in biology where a lot of the initial work on allosteric regulation was actually originally done. And so hemoglobin is a molecule that includes 4 individual polypeptide subunits that are shown here, 2 alpha subunits and 2 beta subunits. And these come together because there are very strong binding interactions between these subunits that hold them together. In fact, this is such a stable complex that even when you add a chemical denaturant like urea, this thing actually stays together. And each subunit at these interfaces are located these heme groups. And these are the groups that actually bind to oxygen and carry oxygen in the blood. And so it turns out that hemoglobin exists in 2 major conformations, called either the T state or the 10th state, or the R state over here. And it's really only the R state that has a high affinity for oxygen, although it can bind to the T state just with much lower affinity. And the cool thing about this system is that when hemoglobin is in the T state, like over here, so it has a relatively low affinity for oxygen, if oxygen does bind to one of the heme groups in one subunit interface of this complex, then that actually triggers a conformational change in the rest of the subunits that increase the affinity of binding for oxygen by a lot. Okay? And that's classic allosteric regulation. So binding in one place on this complex affects the affinity of interaction of substrates, in this case oxygen, on all the other sites. And so it becomes a much higher affinity complex and has the ability to bind to oxygen much more tightly when it's in the state over here. And that's actually a critical property of hemoglobin to be able to do that, because it means that this is a molecule that's very well designed or evolved to bind to oxygen very tightly and in this case cooperatively under conditions where it needs to do that, and then it can also release oxygen into tissues where the concentration of oxygen is very low. Okay, so that's classic allostery. And cooperativity here related to allostery, and I think we're just about done. This is just allosteric regulation. We can maybe we'll just talk about that for a couple minutes on Monday. Have a great weekend, folks.